COVID-19 patients who develop severe disease experience cytokine storm, which is frequently fatal. The mechanism of cytokine storm is poorly understood and development of new therapeutics is urgent. Now, several promising clinical trials are underway, but of course success is not guaranteed. So in order to address this challenge, we're developing a data-driven mathematical model of cytokine storm as observed in COVID-19 patients. So this allows us to not only understand mechanisms, but also to predict best protein targets for drug intervention. Importantly, our process is calibrated to ensure that protein targets emerging from our network model are actually druggable. To overcome issues with proteins not exhibiting obvious binding sites, we implement novel protein dynamic simulations to expose what are known as cryptic allosteric sites. So to construct the initial model, we built a repository focused on those cytokines most prominent in COVID-19 cytokine storm, the cells that produce them, the receptors and target cells. Then using mathematical modelling, we explored our repository for cytokine interactions and the statistical properties of the model. So we've calibrated the first version of the model using publicly available data from COVID-19 patients. Now our preliminary analysis has unravelled potential targets for therapeutic intervention and importantly directions for further investigation. So one such target, IP10, has been recently demonstrated to be well correlated with viral load and to loss of lung function in COVID-19 patients. So we've been able to probe the protein structure of IP10 using publicly available data and run initial simulations to identify potential druggable sites which should provide opportunities for drug repurposing. Building on our results achieved during this hackathon, we're now going to turn our focus to building and developing new collaborations with experimental researchers and medical doctors in the field. We believe that integration of new data will help us to expand and better calibrate our models, and we are really confident that we will reveal new targets, providing new drug repurposing opportunities to ultimately improve COVID-19 patient outcomes.